Hi guys, welcome to my office. I'm gonna do a quick, raw, unedited, beautiful video of pieces that I wasn't able to cover in lecture this week with you all. So, one of which we talked a little bit about the oral cavity, right? In terms of a key function, it's just mechanically, physically breaking down food. We also have enzymes in our saliva that are going to work to break down our carbohydrates, to begin to break down carbs, to begin to break down lipids. But recall that we don't get any protein chemical digestion here. We're just breaking it down from big pieces to small pieces. If you look on the study guide for lab, you do need to be able to identify the incisors that are going to cut, the canines that are also going to that pierce or tear, and then certainly the premolars and molars that are going to work to mainly grind your food and allow you to swallow what we will call a bolus. Another important thing to keep in mind with saliva, we make about 1 liter a day on average. That is a lot when you think about it. Here are the glands. I have a video of the models that I'm going to post in the next day or two here. This is the parotid, a huge one just underneath your masseter. That's what's here. These are exocrine glands, so they're going to have ducts. So here's a duct carrying that saliva into your mouth. So you've got the parotid, you've got the submandibular, and then you've got the sublingual here underneath the tongue, and those are all going to release saliva. Release is controlled by your autonomic nervous system. And specifically, if you recall, you've got your sympathetic branch and you've got your parasympathetic branch. The digestive system, rest and digest, that's the one that's associated with the parasympathetic system. And you'll see in the video that I have posted about regulatory controls of digestion that the vagus nerve, cranial nerve 10, is the only one that extends past your neck, coming from your cranial nerves. And it plays a huge role in regulating much of your entire GI tract. So if you've ever experienced dry mouth when you're nervous, right? Cotton mouth. We've all been there, right? What that is, your sympathetic is dominating over your parasympathetic, and you stop making as much saliva. Saliva also has a lot of antimicrobial properties that help to prevent decay in our teeth. A lot of people think that saliva is on the acidic side of the pH scale, and it's not. It's a little basic. It's a little bit more alkaline. It's fairly close to neutral, but it's not acidic, right? That acidity would wear at the enamel in your teeth. So yes, there are enzymes, but they like to operate in a slightly basic or alkaline environment. If you did that 111 lab where you were collecting saliva and then looking to see how you can manipulate the effectiveness of that amylase, exact same thing, right? It's going to be more basic. So for all sections, I was able to talk predominantly about the stomach and the small intestine. For some sections, I got through a little bit further than others, but this is important, right? What's important are the imports, right? We know that the small intestine itself, it is able to release intestinal juices. I know that's a lovely term, but it basically is just referring to the liquids, water, aqueous solution that's going to help to provide a solvent for digestion to occur on. And then you're also going to have a little bit of alkalinity. So you're going to have some secretions that are going to be more alkaline in nature, also coming from the intestine. Don't forget about the brush border enzymes. So on the villi, you've got the microvilli, and released from those microvilli are enzymes that are going to help complete carbohydrate digestion and help complete protein digestion. And that's all coming from the small intestine itself. But right, we are going to be dependent on the pancreas, the gallbladder, and the liver for additional imports. So you're seeing everything coming together here. This is the duodenal papilla. You've got the pancreatic, hepatopancreatic ampulla right here. Oh, there it is. It's actually labeled hepatopancreatic ampulla. So that is a place where you see a broadening because you have two ducts joining. That's a mouthful. Hepato just means liver. It's always referring to the liver, and then pancreatic. So we're saying, hey, we've got a tube coming from the liver, we've got a tube coming from the pancreas, and this is where it broadens to allow for the secretions here into the duodenum. Endocrine versus exocrine function of the pancreas, both are going to be relevant to quiz one, right? The endocrine function, it's your insulin, your glucagon, working to regulate the homeostasis, blood sugar, right? Don't forget which one raises, which one lowers, if you need to look back in those notes. So the liver's role in digestion. The liver plays so many roles in the body in helping to detoxify and helping with metabolism and helping to package fats up into LDLs and triglycerides and what have you. In terms of its role with digestion specifically, taking a large molecule and breaking it down into smaller parts so we can absorb it. When you consider that piece, its main role is in producing bile. Again, I try to emphasize this in our classes, but not chemical digestion. It is not an enzyme. We're dealing with physical digestion, taking large fat molecules, breaking it up into smaller ones. Bile has a lot of things in it, including phospholipids. So this is going to allow the bile to bind simultaneously to the fat, the nonpolar pieces, as well as to the polar pieces, the water. Because our body is mainly water, right? Our digestive system, we've imported all of this water as we move through the GI tract. And so bile's slick because it can bind to the fats and the water and be able to pull the fats apart. Soap also works this way, right? It binds to the greases and the oils on our clothes, but it also has to simultaneously bind to the water so it can be washed away. So that's the cool thing about phospholipids. Let's keep going. I added this slide, and I know I noted this in class, but these are vessels. These are veins coming from the mesentery, right? That's that double layered serous membrane that leaves your small intestine. You also have mesocolon, which is another type of mesentery that, as you might imagine, is going to leave the colon. And that's actually the colon that you're seeing. The small intestine has been stripped away. But what I want you to appreciate is that when your carbohydrates in all their monosaccharide forms cross the small intestine into the capillaries of the villi, you with me? And then same thing with the amino acids from protein breakdown. When those smallest monomers cross into the capillaries, they head straight through the hepatic, straight up into hepatic portal circulation, right? It goes, the next stop is going to be the liver. With the fats, recall that they are too big and they are too lipid or fat soluble to be taken up into our bloodstream at the level of those villi. So instead, what they have to do is they have to go into the lacteal of those villi. Those are larger lymphatic vessels that have the ability to take up those fats. And from there, it goes in lymphatic circulation, and then it goes to the liver. Okay, so a couple of extra steps that we see with fats, just because they're not polar. We can't bring them into the bloodstream very easily. I talked about this in many classes, and I had a chance to draw the picture on the board, but here are the bile salts breaking down the fat globule into smaller molecules. What is actually absorbed into the lacteal, the term that we give those molecules is right here. They're called chylomicrons. Gallbladder, I addressed this in another video, but CCK, that is the critical hormone that triggers the gallbladder to contract and that bile to be released. If there's no fat, no protein also present in the GI tract, you usually don't get that bile released, or certainly not as much of that bile released. Over here, you're seeing just an example of a gallstone that blocks the ability of that bile to come in. In this case, it's also blocking the pancreatic duct. So you can imagine that digestion for this individual is severely impaired, not able to take in nutrients because they're not able to digest them before they try to absorb them. The large intestine, just big picture stuff here. Note the large intestine is the same thing as your colon. So, but it's going to include some additional divisions. So with your large intestine, you have the ascending colon, the transverse colon, descending colon, This little bit here where it comes back up, that's your sigmoid colon. And then this piece here is going to be the rectum. The key functions, sorry guys, the key functions. So ideally we've absorbed all of our nutrients, all of the calories that we need from carbs, proteins, fats. Ideally, we've taken that all in the small intestine. And by the time we get here to the large intestine, we're really focused on reabsorbing any water that we need and also electrolytes. If food passes, let's say there's irritation in the colon and food passes too quickly, you may get diarrhea because there's not as much time to reabsorb water as that chyme moves through. Similarly, if things move too slowly through your colon, if you don't have enough roughage or fiber in your diet, then you can end up with constipation. Too much of that water has been reabsorbed. Other goals compact any indigestible food, right? And that does include largely those insoluble fibers, and most of which we get from vegetables and fruits. But all of that's critical to feeding this lovely population of bacteria that we store in our bodies. And when those bacteria can feed off of those indigestible carbohydrates, really is what they are, it releases vitamins. And so you do get a little bit of vitamin K. That's really the only nutrient that we get from our large intestine, from this stage of our GI tract. And then certainly another function is going to be to store fecal matter before we defecate.